Okay, so good afternoon, everybody, also from me, and welcome again to the neuroinflammation session. And before starting, I want to, I'm Stefano Giari, I'm one of the chairperson of the session. And before starting, I want to remind you a couple of things. The first one, please, if you have any question throughout the whole duration of the session, ask the question in the question and answer box on the bottom of the screen. And of course, in this case, the questions will be made all at the end of the session. So if you want to ask a question to a specific speaker, please write the name of the speaker and then the question. Otherwise, we'll not be able to assign the question to the speaker. The second time, this is for the speakers, please be in time. We don't want to be too late at the end of the day. So you have your time, please, again, be in time. Now, before starting the actual session, I want to introduce you to my co-chair, who is Michela Campolo. Michela is a young member of the Italian Society of Pharmacology, SIF, and she will introduce to you SIF and how you can collaborate with them. So please, Michela.
Good afternoon. So thanks, Stefano. And we said, I'm Michela Campolo, and I'm a member of the study committee of under 38 member of Italian Society of Pharmacology. So I'm gonna share just some slides to present the society. Okay, so if everybody can see, Italian Society of Pharmacology aims to promote a disseminate pharmacology and its application in Italy and abroad, fostering the drug scientific value and appropriate use. But how the society do this? By supporting graduate students that desire to work in the field of pharmacology, and this by practice or by fellowship, by promoting knowledge in preclinical and clinical areas, by facilitating knowledge exchange and research meeting. Every year, so the society have the National Congress and different workshops. And also by giving its scientific and cultural skill to health authorities and general public in order to contribute to drug policies based on scientific choice. So SIF, the Italian Society has a specific organizational structure that with a president that coordinates the study committee. Inside the SIF, we have a division on clinical pharmacology, a communication area, and a different working group. Actually, the president is Professor Giorgio Racagni. Moreover, we have different external partners, such as AIFA or Famindustria on Scientific Society and different pharmaceutical industries. The under 38 SIF group includes junior and senior members under 38 years old. It was born with the aim to create a bridge between the under 38 members and the SIF board of directors, supporting and promoting initiatives. Actually, the committee is constituted by our coordinator Jacopo Angelini, me, Elisabetta Cagliazzo, Ambra Grolla, and Rossana Roncato, where our advisor is Professor Armando Genazzani. The members have different degrees that go from biology to medicine and have different qualifications, such as PhD or specialty. As well, they cover a huge area of research areas that go from preclinical to clinical and translational ones. Which are the objectives of the under 38 young members? For sure, are to support the younger people professionally that want to professionally grow inside the society. Develop partnership with the youth groups of other scientific societies. For example, recently we organized a webinar about regulatory tools actively contribute to the initiative proposed by CIF, and strengthen the sense of identity of the group and the spirit of belonging. For this reason, we, for example, set up a Facebook group where every member can share protocol, can share a scientific article, or can share new position on fellowship. And annually, every year, and these years will be March 8, 2021, there will be the seminar of pharmacology, where PhD students, fellow, postdoc, and specialist trainers go together to interact and discuss about pharmacology. This will be followed by the National Congress of the Society. That will be from 9 to 13. So just a few things. Thanks for attention, and let's start with the neuroinflammation session. I'm gonna give the floor to Stefano.
Okay, thank you, Michela, for this nice introduction of CIF. So if you're interested, just contact her and she will give you even more information. So now we can talk, we can start with the talk. So the first one is from Simona Schiavi, and she will talk about non-invasive quantification of inflammation, axonal and myelin injury in multiple sclerosis. So please, Simona.
Good afternoon, everyone. And first of all, I'd like to thank you for the introduction and for giving me the opportunity to present my work at this Congress. This study was conducted at the Icahn School of Medicine in Professor Matilde Inglese laboratory and in collaboration with the Washington University in St. Louis and the University of Genoa. Multiple sclerosis is a chronic inflammatory disease of the central nervous system affecting mainly young adults. It is characterized by a broad spectrum of pathological processes, including inflammation, demyelination, axonal loss, edema, and remyelination. Having a biomarker of these very different processes might help in monitoring the different aspects of the tissue damage underlying the progression of the disease. Diffusion basic spectrum imaging, or DBSI, is a magnetic resonance imaging technique that has been recently developed to characterize the water diffusion properties associated not only with axon and myelin injury, but also with hypercellularity. Its pathological specificity has been validated in experimental models, post-mortem tissue sample from MS patients, as well as in vivo healthy and pathological brains, with promising results in terms of quantification of microstructural damage. Thanks to the DBSI sequence and related mathematical model, we are able to compute maps of fiber fraction reported here, which represents the apparent axonal density. Restricted fraction reported here, which reflects water inside small cellular compartments. Non-restricted fraction, which reflects free water and tissue destruction, and this is why it's very high in correspondence of the CSF. Axial diffusivity, or AD, reported here, which reflects the diffusivity along intact fibers. Radial diffusivity, reported here, which is an indirect measures of myelination, and fractional anisotropy, which reflects the fiber integrity. The aims of our studies were to determine the feasibility of DBSI in multiple sclerosis patients at very ultra-high field in vivo, a 7 Tesla scanner, and to investigate the pathological substrate of tissue damage in lesions and normal-appearing white matter. We enrolled 43 patients with clinically defined MS. All patients underwent MRI scanner as well as neurological examination with assessment of motor and cognitive disability through the expanded disability status score and the symbol digit modality test, SDMT. For comparison, 21 age, sex and match healthy subjects were also enrolled and served as control group. When looking at the demographic and clinical features reported in this table, no differences were present between the groups in terms of gender distribution, disease duration, and SDMT, while significant differences were observed for age and ADSS score. On a Siemens Magneton 7 Tesla scanner, we acquired for each subject T1 and T2 weighted images to detect the lesions, as well as DBSI sequence to investigate the microstructure. The identification of lesion type and quantification of the volume was performed in each patient on both T1 and T2 sequences by a single experience unaware of subject identity. In a second step, we use an in-house algorithm to automatically classify each lesions as T2 hyperintense and T1 isointense lesions, which are reported here in green. T2 weighted hyperintense and T1 hypointense, shown in pink here and here. And finally, the most severe lesions, which are T2 weighted hyperintense and T1 black holes, and are reported in blue here. When comparing the different lesion types in terms of DBSI metrics, we used statistical analysis corrected by age and gender. A significant differences between T1 isointense lesions and black holes for all DBSI derived metrics were observed, indicating more severe tissue damage in black holes and higher cellularity in T1 isointense lesions. Likewise, there were significant differences between T1 isointense lesions and T1 hypointense lesions for all DBSI-derived metrics, except for fiber fraction, that suggests that although the tissue damage is greater and the cellularity is lower in T1 hypointense lesions, the overall fiber density is not different. Finally, T1 hypointense lesions differed from black holes in all DBSI-derived metrics, except for the restricted fraction and the fractional anisotropy, suggesting low cellularity in both lesion types. For the lesions with volume bigger than ½ centimeter cube, we compare the values of DBSI metrics at lesion center, reported in dark here, border, reported in gray here, and surrounding normal appearing white method, reported in white. We found that all the metrics were significantly more altered in the center of the lesions, reflecting severe tissue injury and loss, while restricted fraction was significantly higher at the lesion rim, reflecting greater cellularity. This is likely related to the higher density of activated microglia at lesion's boundary, described also in previous PET studies. The same results were also obtained when comparing surrounding normal appearing white matter with the center and the border of the lesions, indicating the presence of a gradient of tissue destruction in corresponding to these big white matter lesions. We also compared the DBSI-derived metrics within the corpus callosum normal appearing white matter between the three groups of subjects: healthy controls reported in white, relapsing remitting multiple sclerosis patients in gray, and progressive MS patients in dark gray. We observed significant differences between healthy controls and relapsing remitting MS patients, except for the restricted fraction, suggesting that either the normal appearing white matter cellularity is not increased or is below the threshold of detectability of our method. As expected, significant differences were also identified between progressive MS patients and healthy controls for all the metrics, except again the restricted fraction. When comparing the two groups of MS patients, we found significant differences for all the metrics except for the fiber fraction and the restricted fraction, with radial diffusivity and fractional anisotropy being the most different. This suggests us a more severe demyelination and white matter injury in progressive MS patients. Finally, significant associations were found between the SDMT score and both corpus callosum normal appearing white matter fiber fraction and restricted fraction, as well as between ADSS score and black holes radial diffusivity. To conclude, with our study, we confirmed the specificity of different DBSI metrics to diverse pathological correlates. We employed the DBSI metrics to characterize focal and diffuse tissue damage in different MS phenotypes, confirming their utility as biomarker of tissue-specific microstructural damage. Finally, our results suggest that DBSI-derived metrics we are able to quantify the cellular component of the lesions and normal appearing white matter. And this may help us to understand the interplay between innate and adaptive immune cells and myelin and axonal changes in driving tissue repair or damage in MS patients. With this, I'd like to conclude my presentation by thanking all the institutions and agencies that supported our work, and thank you for listening.
Okay, thank you, Simona. Thank you very much for the great talk and also for being in time. And again, I remind to everybody, if you have any question, to ask the question in the question and answer block. And so we can move now to the second speaker, who is Giulia Chisbani. And Giulia will talk about peripheral cytokines and fatty acid association with neuroinflammation in Alzheimer's disease and AMCI patients, an exploratory study. So please, Giulia.
Good afternoon, everybody, and thank you for having me today and for giving me the opportunity to present my postdoctorate work at the University of Toronto. As you may know, Alzheimer's disease is one of the most common causes of dementia worldwide and often is associated with aging. One of the first symptoms of Alzheimer's disease is cognitive impairment, and some people experience mild cognitive impairment, which is seen as a transition phase from normal aging to Alzheimer. And these subjects with mild cognitive impairment are often more prone to develop Alzheimer's disease later on. Alzheimer's is also a very complex and heterogeneous disease, and neuropathologically, it's characterized by the accumulation of amyloid plaques, neurofibrillary tangles, degenerating neurons, and microglia activation. And the neurodegenerative disorder in the brain appears decades before symptoms. And it's very important to diagnose the disease in time. So there are diagnosis of Alzheimer's relies on different measures, clinical assessment, for example, as well as MRI PET imaging to look at the brain morphology, and others also used peripheral biomarkers. And these peripheral biomarkers, especially the one from blood, are very accessible, although not always specific for the disease, but they've been associated to the neuropathology. So we start looking in the literature, and we could not see any article that was looking specifically at peripheral biomarker association with neuroinflammation. So we start asking ourselves whether peripheral biomarkers could be actually associated and predict neuroinflammation in Alzheimer's disease. And we were most interested in two peripheral markers. On one side, peripheral cytokines, which are released by the immune cells, and they can be both pro or anti-inflammatory. And on the other side, we were interested in peripheral fatty acids, which can be acquired either through the diet or produced by biological processes like lipogenesis. And also peripheral fatty acids can regulate the activation or the inhibition of the immune system. And among the peripheral fatty acids, the most known are the omega-3 and omega-6, which are essential fatty acids. And finally, fatty acids can influence microglia activity. So to answer our question, we conducted an exploratory study on a very small cohort of subjects, either with Alzheimer's disease, mild cognitive impairment, as well as healthy volunteers. And all these patients, all these subjects underwent PET scanning for TSPO. And TSPO is a translocator protein that is expressed on microglia cells. And when it's elevated, is indicative of neuroinflammation. So what we saw is that the Alzheimer's disease group was having an elevated inflammatory response both in the hippocampus and the prefrontal cortex compared to the other two groups. So We then went to look into the plasma samples of these subjects that underwent TSPO PET scanning. And first of all, we look at a panel of cytokines. And among all the cytokines we studied, IL-6, which is a pro-inflammatory cytokine, and IL-10, that is an anti-inflammatory cytokine, were elevated in the plasma of these Alzheimer's disease patients compared to the other two groups. We then went to analyze another panel of fatty acids, and we look both at the saturated fatty acid, monounsaturated fatty acid, as well as the polyunsaturated fatty acid. And in this slide, I'm focusing mainly to the polyunsaturated fatty acids, and more specifically to the omega-6 group, which is in the upper row, and to the omega-3 fatty acid that is in the lower row. And omega-6 fatty acids are usually associated with pro-inflammatory activity, while omega-3 fatty acids are usually associated with anti-inflammatory activity. So the overall omega-6 fatty acids were modulated, as you can see here, only in the MCI group compared to the healthy volunteers, while the omega-3 were not modulated in these three groups. What was really interesting, though, was that among the omega-6 fatty acid, the arachidonic acid, which is a fatty acid that is always associated with pro-inflammatory activity, and is also the precursor of many lipid mediators associated with inflammation, such as prostaglandin, were modulated between the MCI and Alzheimer groups. The other really interesting fatty acid that we observed that was modulated was the DHA group fatty acid, and we saw that was modulated in the MCI group compared to the healthy volunteer. So the next step was to look into association between those peripheral markers with neuroinflammation. And surprisingly, the cytokines were not associated with the neuroinflammation in any of the groups analyzed. But we observed that some fatty acids were associated with neuroinflammation just in the MCI group. And in particularly, we saw that the saturated fatty acid here and the monounsaturated fatty acids were negatively associated with neuroinflammation, but not the polyunsaturated fatty acid. And with polyunsaturated fatty acids, we mean the total polyunsaturated, including both the omega-3 and the omega-6. We also dissected a little bit more the association, and among the saturated fatty acid, we identified palmitic acid as negatively associated with the neuroinflammation. Among the monounsaturated fatty acid, we found oleic acid, and looking more specifically into the pool of polyunsaturated fatty acid, we identified arachidonic acid as negatively associated with neuroinflammation only in the MCI group. So going back to the initial question, can peripheral biomarkers be associated and predict neuroinflammation in AD? Our study allow us to determine that TSPO PET scanning can distinguish between Alzheimer group compared to the other groups, but our peripheral biomarkers were not able to predict neuroinflammation in a reliable way. But this is still a very relevant study because others reported that both fatty acid and cytokines could be predictive of cognitive function, and because neuroinflammation, chronic neuroinflammation can be deleterious for the Alzheimer brain, it's very important to find easier and more accessible tool to predict neuroinflammation, as PET scanning can be expensive and invasive for these patients. And on another note, our study also opened another question: can fatty acid be a biomarker for Alzheimer? So we looked in the literature and we only analyzed articles reporting fatty acid in Alzheimer's disease and healthy volunteers. in the peripheral compartments. And we observed a great variability in the results. And here I'm just reporting the DHA levels between the Alzheimer group and the healthy volunteer. And as you can see, the results are really variable and there is no consistency. So to conclude, our study requires warrant more analysis, but also warrant a better standardization of sample type, the method used to analyze the samples, but also to increase sample size. And finally, I would like to thank the laboratory of Dr. Basinet and the laboratory of Dr. Misrai and the founding agencies. Thank you.
Okay, thank you again, Giulia. Great talk, great results. And so I leave the microphone to Placido Iliano for the BraYn Educational Symposium. Thank you.
Hello, good afternoon, everyone. I hope you guys can hear me well. So now I'm going to introduce you along with Giulia D'Arrigo for this first educational symposium of today and of the whole conference. So just a shout out to us and to all the brainiacs that are behind the scenes now working to make this happen along with everybody else. So this symposium will work. It will be a session of 2 different speakers. And as it was reminded before, please keep the questions if you have any for the very end of the whole session. So I am honored to introduce you now to Andrea Paulillo, and with a presentation with a talk named Merck Heritage and Commitment in the New MS Era, Past, Present, and Future. So I hope you guys enjoy, and I'll see you later.
Hello, everybody. Good afternoon. Can you see myself and my voice, first of all, just to be sure? Sharing my desktop. Can you hear me? Can I have any confirmation about that?
Yes, yes.
Yes, yeah.
Oh, okay, thank you so much. So, good afternoon, everybody. Thank you very much indeed for inviting me to present, first of all, my now 20 years experience in the pharma world. I'm a neurologist as a background. And I worked a lot in the research field. Just a note, I'm really appreciated the prior presentation about the neuroinflammation Alzheimer's disease because approximately 15 years ago, we tested the interferon beta 1A as a potential treatment for Alzheimer's disease based on its anti-inflammatory property. So my thought in this moment is just to introduce my company, what is our involvement in the last 25 years in multiple sclerosis field. Just a brief introduction on who we are and what is today the real important role of medical affairs. When I started 20 years ago, medical affairs was a sort of on-demand consultant. Today, as you will see during my presentation, medical really plays a strategic role in the development of a medical drug and a strategic role in partnering with all the other departments. And finally, I will show you our commitment in multiple sclerosis. So who we are. Merck probably is one of the oldest pharma company. Is founded by Frederick Jacob Merck. So this is one of the few companies in the head of a family. And 2 years ago, we celebrated 350 years of this company. As you can see, we are approximately 70,000 employees in 70 countries. with a strong investment in a research and development business. So approximately 2 billion euros in investment in research. What should be our ambition? This is our claim, this is our motto. So we would like really to be a vibrant science and technology company. And because science is our main driver, obviously we are working in partnership with many academic centers in try to obtain advancing in genome editing technology, in discovering unique way to treat the most challenging diseases. And particularly so, I know that this is a common word in all the pharma company, really to try to enable the really personalized and tailored treatment for the patient. So these are our main activities. So we work on healthcare, we work in life science, particularly, you know, probably as you researcher, you know that Millipore is one of our company, as well as Sigma Aldrich for, you know, for other activities. But just for your curiosity, also we are, you know, also performance materials. So the majority of crystal liquid for your, for example, iPhone is a Merck product. And this is also the number of companies where are part of the Merck portfolio. So in terms of pharma, so it's always that for us it's extremely important to put patient at the center of our daily work. On the right, you can find our main therapeutic area, oncology, and particularly this new era about immuno-oncology is very amazing new era where really I think we change the story of treatment of oncological diseases. We have neurology, immunology, fertility. We have many products to try to support women that are difficulty to become pregnant, but also at the same time we have a very, we can say, consolidated and classical drug. For example, Euthyrox is one of the product of our company. Here you can find our pipeline. So as you can see, we have our main product for oncology that is avelumab that we are looking at for different indications. So particularly if you have a specific drug, immuno-oncological drug, in this case is an anti-PD-L1 receptor drug, you can test in different potential cancer. But in terms of particularly neurology, obviously we have, as we will see, our main product that is Rebif, that is the oldest one, interferon beta 1A. But we have the cladribine, and but Merck is still strongly committed to MS. As we will see, we have evobrutinib, that is our BTK inhibitor. That we do believe that all the classes of BTK inhibitor will represent really the novelty, the real future for the MS treatment. As said before, medical affair really is becoming crucial. I think so, I would like to say for the success of a pharma company. Often I say when I discuss for the commercial colleague, we don't have to be scared to have a strong medical, because medical close to regulatory represent the most important department for achieving the success of any new drug, and we're close to the market access colleague to guarantee the access for all the patients for any new drug developed by any pharma company. These are our main interaction. As you can see, we have internal stakeholder, we have external stakeholder. It's obvious that so we have a closely relationship, particularly in our company, but in the majority of company, we have very matrix interaction with all the other departments, because we have on one way to guarantee to really to support our customer, in particular physician, to identify the real benefit to risk profile of any drugs, and to give them in a very increasing complex scenario to identify the best position in the treatment algorithm of any drug. And on the other hand, we have to really implement our partnership with academic center for doing research. But at the same time, we have to have increasing relationship also to guarantee the access of the drug to the field, relationship with the payers, regions, and particularly with the regulatory agency, with the IFA, where, you know, there are two commissions, the technical scientific one, where medical is in charge to present any new drug to be discussed, any further commercialization of the drug in Italy. So our commitment to multiple sclerosis, just you know that MS is an increasing and important disease. On one hand, because in the last 20 years, the diagnosis really became earlier. MRI really introduced a big and amazing and awesome change in the diagnosis of this disease. Today, we have approximately 3 million patients affected by MS. You know that there is a gradient from the north to the south. In some countries, we have 1 out of 300 people affected by MS. This is a disease that affected the young patient, and this is in this setting, in this average, is the most disabling disease for multiple sclerosis. And this is unfortunately, like the majority of autoimmune disease, a prevalence of a gender disease. So with the women that are 2 out of 3 patients are female. As far as concerned treatment, I think we have to really have at least 2 main pillars. We have on one end treatment that have an immunomodulator effect, another that have an immunosuppressant one. It serves that the main difference, probably immunomodulators are less effective than immunosuppressant one, but it serves when I have mentioned before the balance between the benefit to risk profile. This is something that we have to take into account. Generally, more efficacious drugs are correlated with a higher risk safety profile. So it's that this is extremely important to try to identify what should be the best balance. We have to take into account that this is a chronic disease. So this patient has to take treatments for at least 30 years in their life. So it shows that today the life on neurologists, I can say MSologists, because, you know, in Italy, we are really like to have a dedicated center for treating MS. We have a center of excellence. Really, Italy represents one of the countries in Europe, but not only in Europe, where the way patients are followed are, in my opinion, and based on my experience, having a relationship also with you know, other countries, Italy is the place where patients are followed really closely and the best way. As you can see, this is what should be the long journey of treatment for MS. When I started 30 years ago, there was interference and then glatiramer acetate. And all over the years, I think that MS is one of the neurological diseases where really there was an amazing changes in the treatment landscape. If we think about epilepsy, stroke, or other diseases, MS is really one of the diseases where pharma companies really did an amazing contribute to generate a lot of possible new treatment and a lot of opportunity for physicians really to have a different, different arrows to try to defeat and treat MS. Again, as said, Merck is completely involved and still profoundly engaged, and we will have some slides about evobrutinib on that. Interferon beta 1A, so just a very short, representation, just to let you know how is long the development of a drug. The first story, the first discussion about using interferon beta is started at the end of 1917. And you see, so there is a long-lasting development of this kind of drug and test hypothesis, because at the beginning for MS was tested, that we are interferon beta 1A, interferon alpha, we have interferon beta 1B, interferon gamma, different type of interferons. So before joining to the importance of the two interferon beta 1A and B, there was a long research around. So the availability of the drug arrived at the end of 1999, and with different evolution. And when a drug is commercialized, it's never finished the story for a pharma company, because there is the so-called life cycle management of the drug, and that means to try to have eventually a new formula that can improve, for example, the quality or the pureness of the drug, but also considering that this is a drug for chronic disease. What is extremely important is to try to identify tools for self-injecting that may improve the quality of life of the patient. For this reason, as you can see, many self-injecting devices were developed by Merck and also by other companies. Interesting one is really an electronic self-injecting device that may also score and quantify and upload into a cloud the adherence of the patient to the drug, because as you know, adherence is strongly correlated to the efficacy of any kind of treatment. Treatment low adherence or treatment discontinuation is strongly closely associated with also with the progression of disease and the outcome of the disease. And as we said, interference is a pleiotropic drug. It started as an antiviral drug, but it may act in different key point, in different signaling activities, moreover in balancing the pro-inflammatory, anti-inflammatory activity, but also in reducing, for example, the chemoactive activity of macrophage that may permit the disruption of blood-brain barrier and the entering of lymphocytes into the brain during the activation of the signaling the inflammation. Cladribine tablet is the second most important achievement of our company. Is an interesting drug because the study started also when it was identified studies 2 young girls affected by severe immunodeficiency. and related to a really strong lymphocyte reduction. This was identified in a genetic deficiency of adenosine deaminase. And this is this ADA deficiency was shown to cause the accumulation of the osteoadenosine. So cladribine was designed again at the end of 1987, was studied in the US, and it was designed by substituting a chlorine atom for hydrogen atom on the osteoadenosine. So this may permit to make it largely resistant to degradation by the adenosine deaminase enzyme, allowing cladribine to preferentially target the most active replicating cell like lymphocyte B and T, inducing an apoptosis phenomenon. So in this way, cladribine is really able to determine an interruption through this induction system, a reset of the immune system, and generating a repopulation of lymphocyte B and T that in some way are less reactive and less inducing an autoimmune aspect. The other important, extremely important aspect of cladribine, as said, we are talking about a disease lasting at least 30 years in a MS patient, is the posology. This is one of the drugs that is the only drug that this is weighted for the weight of the patient, so it's 3.5 milligram per kilos. And so any patient may intake 5 to 10 pills in 2 weeks. This is the first month of treatment. Or 5 to 10 pills the second 2 weeks. This is and then this is the first year treatment. The same 10 days of treatment in the second year, and this may cover year 3 and year 4, because 75 of the patient may not present a new additional lesion or activity into the brain. So it shows that this kind of posology, looking at the quality of life of the patient, really represents an important achievement for the quality of life of the patient. And say this is extremely important to distinguish treatment that continuously determine immunosuppression to the treatment that generally are partially administered. It shows that non-determine if the effect is long-lasting after this kind of pulse induction. These are the registrative study, say a long-lasting story before going the access to the patient. Today, what is extremely requested by authorities to put in place the so-called past study, so to look at over a longer period of time in the real life, the safety profile of our drugs. Finally, evobrutinib, because as said before, there are now 18 drugs available for treating MS in the landscape. But if you look at this slide is represented the so-called NEDA, no evidence of disease activity. That means no new lesion in MRI imaging and no new clinical activity, relapses or disability progression. As you can see, the most efficacious drugs are able over 2 years to have just half of the patient without progression of the disease. So this is important because it means that are needed new mechanism of action that are able to treat MS and to be added to the armamentarium of our neurologists. So evobrutinib is one of Barton tyrosine kinase inhibitor. As you can see, you can find a dual mechanism. It's able to work on...
Sorry, Andrea, Paolillo. Sorry.
Finish
if you can
the
close.
time.
Thank you. Yeah, if you can close. Conclude.
Okay. This will be, it's also the colleague after on will speak about that. So dual mechanism of action, but what is extremely important, may work on different aspects in second signaling. And so we really do believe that will represent the future of MS treatment. Just a final slide, you know, during this pandemic period, the importance to find the treatment for COVID-19, Merck really have an amazing program to support in different trial the use of interferon beta 1A for treating the pro-inflammatory cascade. And also what is extremely important, these are global studies, but importantly, also Italy is providing an important support with the Intercop study at San Raffaele Hospital, another study starting now with the support of the Institute of Superior Health. Thank you so much.
Okay, thank you. Thank you, Andrea Paolillo, for your presentation. And now I'm glad to present the next speaker of our BraYn Educational Symposium. And she is Francesca Sangalli from IRCS Ospedale San Raffaele of Milan. And she will present a symposium entitled Innovation Through Patient Needs, BraYn Tyrosine Kinase Inhibitor Development Journey. And she's an invited speaker from Sanofi. So I give you the word to Francesca Sangalli.
Thank you. Okay. Can you see my desktop?
Yes, we can see.
Okay.
Yes.
Okay. Good afternoon to everyone. I wanted to start thanking for the opportunity to speak about this interesting topic. I'm a neurologist at San Raffaele Hospital and I work in the Multiple Sclerosis Center. Sorry, I have a problem going on with my slides. Okay, these are my disclosures. BraYn tyrosine kinase belongs to the TAC kinases family. It was initially identified as the defective protein in human X-linked agammaglobulinemia. And now it is known to be predominantly expressed in hematopoietic cells, such as B cells, macrophages, and mast cells. BTK is an important kinase in the B-cell receptor signaling pathway. Particularly, it links the B-cell receptor to NF-kappa B activation and translocation into the nucleus, where it promotes gene expression, which is important for survival, proliferation, activation, and differentiation of B cells. BTK is also important in B malignancies since its aberrant activation is involved in both leukemia and lymphomas. Its expression was identified in cells of mantle cell lymphoma and chronic lymphocytic leukemia. And the B-cell receptor signaling pathway plays also an important role in B-cells lymphomas. Therefore, BTK inhibition can afford a promising clinical benefit in B-cell lineages malignancies. Ibrutinib was the first-in-class BTK inhibitor approved by FDA in 2013. It was able to induce high overall response both as overall survival and also as progression-free survival in refractory relapsed MCL and CLL. It also showed an acceptable tolerability profile. It is also under evaluation for other indications in B-cells malignancies, either alone or as combined therapy. Since ibrutinib approval, many academia and industry efforts have been made in order to produce molecules with a better selectivity in order to reduce collateral kinase inhibition and so to reduce side effects. Research focused both on irreversible and reversible inhibitors, where irreversible inhibitors might provide higher efficacy, while reversible inhibitors are believed to have better safety profiles. And it is to note that most clinical trials are involves irreversible inhibitors. Besides these researches, BTK inhibitors are now gaining importance with new indications, especially for autoimmune diseases such as multiple sclerosis. Indeed, B cells in multiple sclerosis have now a role which is central for pathogenesis and propagation of disease. In fact, historically, B cells were considered important for their ability to differentiate in antibody-secreting plasma cells, which provided the diagnostic hallmark of oligoclonal bands produced intrathecally. Now we know that B cells have many other roles, especially B cells expressing B cell myelin reactive B cell receptor can internalize, process the antigen and present it to T cells, thus stimulating their activation and proliferation. They can also produce pro-inflammatory cytokines and chemokines, which can create an inflammatory environment. And they are also important for differentiation of follicular dendritic cells and for the establishment of tertiary lymphoid organs, which are important for activation and proliferation of autoreactive cells. The role of the B cells in multiple sclerosis were confirmed by the highest efficacy of anti-CD20 therapies, especially of ocrelizumab, which was highly effective in relapsing remitting MS, and which also was able to reduce disability progression in primary progressive MS. Despite this success, there is the problem of prolonged pan B cell depletion, which can provoke an higher risk of infections due to impaired humoral responses, especially considering that MS is a pathology usually diagnosed in young people and that requires prolonged therapy. Also, pan B cell depletion can reduce subsets of regulatory B cells, which would be favorable in multiple sclerosis. So we need a different approach to B cells. And in this inhibition of BTK can interfere with B cells activity. It can reduce B cell receptor mediated activation, and it can also reduce expression of molecules such as MHC class 2 and co-stimulatory molecules involved in antigen presentation and activation of autoreactive T cells. Moreover, BTK is more expressed in differentiated and class-switched B cells, which are the key players in MS pathogenesis. So we can target directly pathogenic B cells while leaving resting B cells and plasma cells to their controlling function and their protective function. Moreover, BTKs are not expected to cause B-cell depletion, and so we can restore function of B-cells within days after drug suspension, for example, in the case of an adverse event. Besides the important role of BTK in B cells, it is also important in myeloid cells, since it mediates Fc receptor and Toll-like receptor signaling, and so can activate and promote maturation of myeloid cells. And it's also important for the regulation of the inflammasome, which is the key inflammatory machinery of the innate immune system. Myeloid cells are important in MS pathogenesis, both activated monocytes and macrophages, which migrate into the central nervous system from periphery, and resident myeloid cells, the microglia. The activation of myeloid cells in MS pathogenesis is of the subgroup of the so-called M1 activation, which can be very damaging to the central nervous system. It can determine direct damage to oligodendrocyte, and it can, myeloid cells can act as antigen presenting cells and also produce cytokines important for activation of other inflammatory cells. This activation of myeloid cells is therefore important in active lesions, which are the hallmark characteristic of relapsing multiple sclerosis. But the microglia is also important in the so-called smoldering lesions. In this lesion, we see an inactive center, but a ring of activated microglia. And this lesion can cause great damage to the central nervous system, especially in progressive patients, and they are now considered as important drivers of progression in multiple sclerosis. So we can imagine a role of BTK inhibitors also in controlling myeloid cells, since BTK inhibitors might reduce activation and maturation of B cells, and they can also cross blood-brain barrier, so they can not only act peripherally, but they can act directly into the CNS. So we can imagine two targets, a new lesion in relapsing multiple sclerosis and the evolution of smoldering lesions in progressive multiple sclerosis. For these reasons, BTK inhibitors are now tested in the treatment of multiple sclerosis. We have two phase 2 trials. One is the trial with evobrutinib, which was tested into 3 different doses against placebo. And the dose of 75 milligrams daily demonstrated the ability to significantly reduce gadolinium-enhancing lesions measured at 12 and 24 weeks of treatment in comparison to placebo. There was no difference in annualized relapse rate and disability progression, but we must consider that 24 weeks is a very brief time of observation for multiple sclerosis. Tolebrutinib was also tested into 4 different doses, again against placebo, and the 60 milligram dose daily was able to reduce both gadolinium-enhancing lesion and the formation of new lesions into 2 sequences at 12 weeks. So based on what we told about pathogenesis and this result, phase 3 trials are now starting in MS. We have many trials, especially we have 3 trials in relapsing multiple sclerosis where BTK inhibitors are tested against teriflunomide. We also have 1 trial in secondary progressive MS, where tolebrutinib is tested against placebo, and 2 trials in primary progressive MS, 1 against placebo and 1 against ocrelizumab. Concluding, inhibition of BTK is an evolving approach of interest in neuroinflammation. BTK inhibitors have the capacity to assess and accumulate in central nervous system, so act directly where pathogenesis expresses itself, and they can be active in both B cells and microglia, which, as we saw, are key players in MS pathogenesis. It can be so a new treatment option for both relapsing and progressive MS. Perhaps we cannot expect similar results of depleting therapies, so we can think of BTK inhibitors as an option for low to moderate activity patient, but we can also imagine consequential treatment for ugly active patient. Perhaps we can start with a B-cell depleting therapy in order to induce control of the disease, and then start with a BTK inhibitor, which can regulate the activation and proliferation of B cells while allowing repopulation. And also we can imagine to use BTK inhibitors in progressive MS, and their ability to act against microglia are very important because we can act directly against the pathogenic effectors of smoldering lesions, which are very damaging, which mediate progression, which is still an unmet need for our patient. Of course, we will need further research in order to assess the efficacy and safety of these drugs in a larger population. Thank you for your attention.
Thank you very much, Francesca, for your presentation. And now I want to close the first educational symposia, and I will give the word to the chair of the neuroinflammation session.
Thanks, Julia. So, and welcome back to the second part of neuroinflammation session. I'm pleased to present the Dr. Adriano Lama with his presentation, Converging Mechanism of Palmitoyl ethanolamide on Meta-inflammation and Neuroinflammation, Relief of Anxiety-like Behavior in Obese Mice. Thanks.
Good afternoon to everyone. I will share my presentation. Okay. If it's okay, I can start. Yes,
we see.
Okay. Good afternoon to everyone. First of all, I want to thank the BraYn committee for the great opportunity to show my data about the effects of palmitoyl phenolamide on neuroinflammation and anxiety-like behavior in obese mice. Obesity is a disorder characterized by a chronic low-grade inflammation termed as a metabolic inflammation or meta-inflammation. This low-grade inflammation impacts not only metabolic function, but also on CNS, where the lacking or the missing blood-brain barrier integrity can lead to neuroinflammation in many brain areas and triggering the development of neurological disorders. Indeed, free fatty acids reach the hypothalamus, and whose alteration can lead to an impairment of neuronal circuitries of other brain areas, including hippocampus, triggering the development of neurological disorders such as depression, anxiety, and memory deficit. Palmitoyl phenolamide is an endogenous fatty acid amide that belongs to the family of N-acyl phenolamines and originally associated to its congener anandamide. Palmitoyl phenolamide owns well-known central effects through many different mechanisms of action, including the activation of PPR-alpha. Our recent work has demonstrated the metabolic effects of palmitoyl phenylamide in obese mice. But on the other hand, we also demonstrated the antidepressant effects of this compound in these obese mice. In particular, in this last paper, we have demonstrated that PA limited depressive and anhedonic-like behavior, rebalanced the monoamine system in hippocampus, nucleus accumbens, and prefrontal cortex, and stimulated the hippocampal synaptic plasticity and neurogenesis. So our next step was set on PA capability in limiting neuroinflammation and anxiety-like behavior in obese mice. To this purpose, we used the male black 6 C57 mice at 6 weeks of age that were divided into 3 groups: standard group, high-fat diet group, and high-fat diet that were treated with ultramicronized PA 30 mg per kilos per os die. The treatments started after 12 weeks of high-fat diet when obesity was full-blown and lasted for 7 weeks. At the end of the experimental period, we performed the open field test to evaluate the anxiety-like behavior of these mice, and PA limited the thigmotaxis induced by high-fat diet. The behavioral changes induced by high-fat diet and PA were accompanied by monoamine changes in the amygdala, a brain area responsible for the mood regulation. And PA limited the dopaminergic, the high dopaminergic and noradrenergic hyperactivity of obese mice. Then we also demonstrated the anti-inflammatory effects of PA, limiting the systemic levels of lipopolysaccharides, monocyte chemoattractant protein 1, interleukin-1 beta, and TNF-alpha. Then we moved on hypothalamus, the meeting point between periphery and CNS, where the lacking blood-brain barrier integrity can lead to the translocation of detrimental factors to CNS, especially at the median eminence level. Here, we demonstrated that PA decreased the transcription of pro-inflammatory mediators such as NF-κB and interleukin-1 beta. At median eminence level, we also demonstrated that HFD induced a strong microgliosis and astrogliosis that were counteracted by PA treatment. Then we moved on hippocampus, where the blood-brain barrier integrity plays a pivotal role to avoid the translocation of these detrimental factors from periphery. And on one end, we evaluated the mRNA expressions of tight junction protein, pivotal protein for the blood-brain barrier integrity, and PA increased the transcription of zonurin 1, occludin, and brain-specific claudin 5. On the other hand, we evaluated the albumin extravasation by immunofluorescence analysis as an index of blood-brain barrier integrity, and the PA treatment decreased the albumin-positive neurons in dentate gyrus and CA1 of hippocampi of obese mice. A recent work has demonstrated that mast cells are early activator of LFPS-induced neuroinflammation and BBB dysfunction. So we evaluated the transcription of 2 murin-specific proteases of mast cells, kinase 1 and tryptase beta 2. And PA decreased the transcription of these 2 detrimental factors. As shown in hypothalamus, even for hippocampus and dentate gyrus and CA1 of hippocampus, PA decreased the number of IBA1 positive and GFIP positive neurons altered by HFD. Finally, we also demonstrated that PA decreased the protein expression of toll-like receptor 4 and the transcription of its downstream gene MID88 and the following pro-inflammatory cytokines TNF-alpha and interleukin-1 beta. In conclusion, we can say that PA limited the obesity-induced unsightly neuroinflammation, limiting tegmotaxis, the monoamine balance, and improving blood-brain barrier integrity, modulating the immune cell changes, and limiting the activation of toll-like receptor signaling. This data add a further piece of palmitoyl phenolamide activity for complex disorders, including central disorders induced by obesity. Please let me thank my research group, Pharmeta Lab member of the University of Naples, Yale University, where I spent 1 year of my academic career, University of Foggia and Polytechnic University of Marche for the precious collaboration, Epitech Group for the funding sources, and of course, all of you for your kind attention.
Thanks, Doctor, for the good talk. So we are going to start with the question time. We have few times, so I can do just one question for each speaker. I'm going to start with the first two speakers in the first part of the session, Dr. Simona Schiavi and Giulia Chisbani, if they can open the webcam. Okay, so let's start with Simona Schiavi. Maybe you can reply to the other question directly on the question and answer slot. I'm gonna read just one. Thanks. Okay, so the first one, Simona, okay, is, did you think about correlating some blood markers such as cytokine levels with the number and extension of the lesion you found in the brain? This could give you info on the involvement of inflammation in multiple sclerosis onset and progression.
Okay, thank you for the question. Actually, unfortunately, for the patients we analyzed, we didn't have any blood sample, but this is definitely something we will look at in future, and it's a very nice hint. So I thank Elisabetta Volpe for suggesting this.
Okay, thank you. Okay, I have another question for the Dr. Giulia Chisbani. If I can open the video, I cannot see. Okay, no. Maybe it's not here. Okay, maybe we can move on for question for Sangalli Francesca, and then maybe come back.
Good evening.
Okay, good evening. So, okay, Giulia, maybe you can do the question. And for Chisbani, Giulia, maybe she can reply directly on the question and answer slot. Thanks.
Okay, so for Dr. Sangalli, we have this question from Jose Elefante Canavate that asks, would it be possible to administrate the IBTK intrathecally in order to minimize the secondary effects in the periphery and boost the effectivity within the CNS?
Thank you for the question. I think that intrathecally administration is always aggravated by side effects. So considering that BTK inhibitors can pass through the blood-brain barrier, I don't think it would be necessary. And in multiple sclerosis, perhaps we want to act both peripherally and centrally in the central nervous system. And moreover, I think that to reduce side effects, it would be perhaps more important to produce selective BTK inhibitors more than administer them intrathecally.
All right, thank you for your answer. And then I'll give the word again to Michela Campo.
Okay, thanks. Maybe Giulia is online, so I can do the question.
No, she left the chat.
I'm back.
Oh, okay, okay.
She's back. Okay, just Giulia, yeah. Do you know if your patients carry ApoE mutation or not?
That's a good question. We don't know. This study was not designed to look at the fatty acids initially, but it was focused only to look at the TSPO levels. So our study was a secondary analysis. So that there are some, also we don't know the dietary history of these patients. So our data has to be also taken carefully because the patients we didn't have all the necessary information.
Okay, thank you. So move on with Dr. Lama. If he's here, maybe can open. Okay.
Yeah.
So we just do question. Okay. And this guy writes, I thank for the very interesting presentation.
Thank you.
So we have two questions. What do we know about the actual molecular targets and pharmacodynamics of PIA? And the second one is there any known endogenous metabolic pathway that leads to the biosynthesis of PIA?
For the first question, these are very interesting questions. For the first one, I can see that this is our next focus. Try to explain the molecular target of PA within in vitro approach. Our experience maybe can be with a PPAR alpha involvement, but PA can do it with other molecular target such as CB2 or TRPV1, but this is our next step. And for the second one, palmitoylphenolamide is in on-demand fatty acid that when our body need can synthesize this compound, and after that this PA is metabolized rapidly.
You're welcome.
Thank you. So now thanks to all the speakers. I just would like to thank the BraYn organization that gave me the possibility to present for invitation. Thank you. So let's move to the neuro-oncology session.